Hey, today on Flippin Science, we're looking at DNA extraction and PCR.
Well, it's amazing what heating and cooling and heating will do. PCR, when you need to detect mutation. PCR, when you need to recombine.
Thanks to Sam Moyle for producing this PowerPoint. So science understandings, DNA can be extracted by cells, modern techniques can be used to analyze even small amounts of DNA, and segments of DNA can be multiplied using polymerase chain reaction, PCR, and then you need to describe what PCR is. So DNA can be extracted from cells. You've probably done this already in year 10. We use a combination of shampoo, salt, and ethanol to extract DNA from cells. We do strawberries, for example. To do that, you need to get rid of the cell membrane so that you expose the nucleus, and then you need to get rid of the nuclear membrane, so you expose the DNA. And both of those can be done by shampoo. Shampoo is good at breaking down oils. And then there's other proteins and things to remove, so histones, which are the proteins the DNA wrapped around, other proteins that are in the nucleus. That needs to be removed from the DNA, and then we can isolate that by using ethanol, and then you separate it out into a layer. You can see the layer down here, or you can use it by centrifuging, so you spin it around really quickly and the DNA will sit down the bottom because it's heavier than the other stuff. We're going to go into sequencing DNA in another video. Fred Sanger, he determined a way of identifying the sequences of bases in DNA, but to do that you need to make large numbers of copies of the DNA that you're looking at. So in 4 different test tubes, he heated the DNA so that the 2 strands separate, so the strands are held together with hydrogen bonds that you can separate using a bit of heat. So he added a primer, which is a short piece of DNA that matches the start of the DNA that you want to copy. The primer also prevents the 2 strands from zipping back up again really quickly. Ooh. He then added DNA polymerase, which is the enzyme which makes copies of the DNA. And an excess of nucleotides, so A, C, T's, and G's. He also included a modified nucleotide which would stop the DNA polymerase from working after it reached that nucleotide. What that would mean is that once the process stopped, you knew which nucleotide was the last one added, and then you could analyze the lengths of the DNA strand and determine the nucleotide sequences. So this is one way of sequencing DNA. The PCR is the way that you make many copies of the DNA. So PCR works by, it's basically molecular photocopying, we refer to it as. You can multiply numbers of DNA by orders of magnitude really quickly. It was developed by Kerry Mullis in 1983, and it involves a sequence of steps. So you denature the DNA, so you heat it up to separate the DNA into 2 strands. You've added a whole heap of primer into the mix, and that stops the strands from re-zipping back together. DNA polymerase is added, and the DNA polymerase is added from a heat-tolerant bacteria, and this is important. So the question here is, why is that necessary? The heat-tolerant bacteria, DNA polymerase is an enzyme, it's a protein. It's from a heat-tolerant bacteria, and this process involves a lot of heating and cooling. So your DNA polymerase needs to be able to be resistant to that excess heating and not change shape. If it changes shape, then it won't work. A whole heap of nucleotides, so A, C, T's, and G's added. You cool down the mixture, and the nucleotides have gone in, the polymerase has joined up the DNA, so now you have 2 strands of DNA from your 1. And then you do this many times. So you do it again, you get 4 copies. You do it again, you get 8 copies, and so on and so on and so on. So as long as you have primer, nucleotides, and polymerase present, you can keep doing this for a long time and get many copies. So here's the steps again. So you denature DNA by heating it up to nearly 100 degrees for 20 to 30 seconds. This allows the 2 strands of DNA to separate. You lower the temperature down so that the primers can sit on the DNA, and that stops the DNA from zipping back together completely. You then warm it up so that the DNA polymerase can do its job. Remember, it's from a heat-tolerant bacteria, so it needs to get to the temperature that the bacteria was comfortable at. That allows the polymerase to make a new complementary strand of DNA. You let that keep going, and then you cool it back down again. So then you've doubled it, and then you can just repeat these steps over and over again. So here's a picture showing that. So here's your original DNA that you want to make a copy of. Here's the primer. Here's your nucleotides, your A, C, T's, and G's. You heat it up so the strands separate. The primer attaches to the DNA strands. The DNA polymerase starts making the copy of the DNA and starts working back, and then you cool it down. And then you repeat those steps over and over again. Here's an animation showing this. So you're warming it up, so we're up to 94 degrees, so the strands separate. You chuck on your primer, and then you get your polymerase wandering along, making copies of the DNA, and then you cool it back down again. Today on Flippin Science, we talked about DNA extraction and PCR. That's it for Flippin Science today. See ya.